Phagocytes. Phagocytes are produced in the bone marrow, especially in the big bones of our body. It is produced throughout our life, from birth to adulthood, or until our old age. They are known as scavengers of our body. Phagocytes could be a neutrophil or a macrophage. Neutrophils. Neutrophils are 60 to 70% of our total white blood cell. Their appearance looks like this. This is the diagrammatic version wherein you could see there is their cytoplasm, of course, but this violet area is known as their lobed nucleus. And this is the reason or the structure why they can squeeze through the capillary.
Inside your body right now, white blood cells are moving through your tissues on the hunt for pathogens. But this cell is in a petri dish in a lab in Austria, where researchers have been putting white blood cells through a variety of obstacle courses. These experiments are designed to reveal the secrets of their impressively fast locomotion. And understanding how these immune cells speed through the body could help reveal how cancer cells move and spread. These neutrophils are racing through a maze. They're attracted to a chemical at the other end, and by sensing the chemical gradient, they can pick the shortest path. But the inside of your body isn't like a maze with a choice of uniform pathways. Instead, immune cells need to be able to squeeze through gaps between tissue cells and pick the best route. In a new experiment, this dendritic cell is given a choice of 4 paths of different widths. It probes the different options before picking the widest path, allowing it to move faster than it could through a narrower channel. This ability to sense the size of gaps means that cells moving through the body can avoid dense areas and find the fastest routes rather than just the shortest ones. When trying out different options, these cells position the big, bulky nucleus near the front. If the nucleus fits, the rest of the cell will too. This ensures the nucleus doesn't get damaged squeezing through too narrow a gap. Once the cell has decided on a path, the other protruding probes are retracted using a kind of internal cellular skeleton made of microtubules. If these microtubules are disrupted so that they can't retract, as has been done here, the cell will pull itself apart. Cancer researchers are particularly interested in understanding the mechanisms of immune cell locomotion. Tumour cells may activate these same systems when they start to metastasize, allowing them to move through tissues. So understanding the sophisticated locomotion of white blood cells like these could help scientists stop cancers spreading around the body.
The number of their lobes vary. So they are active throughout the body, so anywhere, everywhere, we can find them. But even if they are found anywhere and everywhere, we have to note that they are short-lived and they are released in large numbers during infection, or especially if you have a cut. Their job is to engulf and digest any pathogen, and that process is what we call as phagocytosis. This one is a photomicrograph using a light microscope, and the violet lobes that you can find in here is still the lobe nucleus. The area surrounding this lobe nucleus would be the cell membrane. There. Next. This is a video about neutrophils. So you could see there the very big thing that's moving. That one is an example of a neutrophil. And the black fly or the black dots that are together are the pathogens. And the neutrophil is now looking for it, chasing it, so that it could eat that particular pathogen. This image in here is an electron micrograph, and in here is taken with a light microscope. The nucleus of this one, let's have this one and this one for example. The nucleus in here are whole, it's not lobed. It's similar to a regular nucleus, but it is just way more massive. Macrophages are larger than neutrophils. They actually live long and they are found in several organs of our bodies. Their job is to cut the pathogen and display the antigen. Again, remember, antigen acts like an ID, and this helps our white blood cells determine if they are our own cells or if they are a cell of a pathogen. These macrophages travel in the blood as monocyte and mature to become a macrophage once they are settled in an organ. This one is a micrograph of both neutrophil and macrophage for us to see or differentiate them or compare them. For example, this one is one neutrophil and this one is a macrophage. This one is way bigger. This one is way bigger than one piece of a neutrophil in here. Okay? This one is not one piece of a neutrophil, rather it is a cluster. You will know that because looking at the nucleus, there are like different lobings, and the lobes of the nucleus is not connected to each other. This, on the other hand, is electron microscopy. Again, there is this several nucleus occurring around, while in this one, the macrophage, the nucleus is just one big chunk. You might be thinking, what is MGG staining? MGG staining stands for May Grunwald's Gemsa stain.